Resilient little thing, just like mama made you. No one needs to save you. Na 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 na. Wildflower in the spring, no they can't contain you. Through the cracks you break through. Na 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 na. I deserve congratulations, 'cause I came out the other side. I've been having revelations, and I'm gonna let them shine. I deserve congratulations, and never thought that I'd survive. If you tell me I won't make it, and that's when I super blue. Super blue, super blue, super blue. Resilient little thing, just like mama raised you, so you got that wildfire in your soul. Don't you ever let it go. Make it burn so bright that they all know. I deserve congratulations, 'cause I came out the other side. I've been having revelations, and I'm gonna let them shine. I deserve congratulations, and never thought that I'd survive. If you tell me I won't make it, and that's when I super blue. Never rained in the desert till I picked me for the very first time. It's darkest before the sunrise. That's when I never rained in the desert till I picked me for the very first time. It's darkest before the sunrise. That's when I super blue, super blue, super blue, super blue. I deserve congratulations, 'cause I came out the other side. I've been having revelations, and I'm gonna let them shine. I deserve congratulations, and never thought that I'd survive. If you tell me I won't make it, and that's when I super blue.
Someday I might miss you, but
up right now. It took me a while to get back on my feet. It took me a while to smile again. It was a shame that I could ever be so weak, but I love you, girl. Yes, I did. Hey, someday I might miss you, but I sure miss you right now. I was blue as a man could be when you left me, but I sure miss you right now. When you put me down, I was all torn up, sure enough, crying every night. Well, now I see the light, and everything feels so new. Oh, I finally got over you. Hey, someday I might miss you, but I sure miss you right now. I was blue as a man could be when you left me, but I sure miss you right now. Some nights I start to feel real weak. I get so low down, I can't hardly sleep. But I know this too shall pass, and at last I don't miss you, baby. Someday I might miss you, but I sure miss you right now. I was blue as a man could be when you left me, but I sure miss you right now. Hey, someday I might miss you, but I sure miss you right now. I was blue as a man could be when you left me, but I sure miss you right now. I like the way you say my name, babe, and the way you wake up on a Sunday, rolling around like two warm animals. Yeah, I hope that you feel the same way. Yeah, I hope that you feel the same way. How much should I be reading into? The way you look at me in your room, and I know that nothing's really permanent. I hope that this doesn't end soon. Yeah, I hope that this doesn't end soon. Ooh, and if it feels right, who's to tell you that it ain't right? And when it feels right, oh my, you know it feels right. Ooh, and if it feels right, who's to tell you that it ain't right? And when it feels right. I like the way you say my name, babe. Yeah, I like the way you say my name, babe. I like the way you say my name, babe. Yeah, I like the way you say my name, babe. Hey. Yeah, I like the way you say my name, babe. Oh, I like the way you say my name, babe. Ooh, and if it feels right.
want you to hold me, come and lay with me. Set aside your problems with me, baby. Save your conversation for the basement. I want you speaking in tongues. You know what they say about those when they come together like we're together. You can't let them slip away. You can't let them slip away. We gotta a good thing together, so unpredictable. But one thing I know for sure, you turn my sixes to nines. Frequently fly. We can't deny, no, it's feeling tonight. I know how we can when I look in your eyes, and you're holding me tight, no, we're in love, you and I. You turn my sixes to nines, yeah. Yeah. You turn my sixes to nines, yeah. I want you to hold me, ride this wave with me. Set aside your inhibitions, maybe. Sometimes when it feels right, maybe it's alright. I want you speaking in tongues. You know what they say about those when they come together like we're together. You can't let them slip away. You can't let them slip away. We gotta a good thing together, so unpredictable. But one thing I know for sure, you turn my sixes to nines. Frequently fly. We can't deny, no, it's feeling tonight. I know how we can when I look into your eyes, and you're holding me tight. We're in love, you and I. You turn my sixes to nines, yeah. Yeah. You turn my sixes to nines, yeah. You turn my sixes to nines. Frequently fly. We can't deny, no, it's feeling tonight. I know how we can when I look in your eyes, and you're holding me tight. We're in love, you and I. You turn my sixes to nines.
The mouth of my suitcase says I'm making a big mistake. I'll pay for it one day, just not too late, too late, too late. Brand new shoes that I can't breathe, big balloons that I can't inflate. A crayon and a blank slate pulls me away, away, away. You're guarded, I'm leaving, so sad to go. I'm leaving my heart in San Francisco. That's hot strawberry blonde, no I'll miss so. I'll send you a postcard and I get where I've gotta be. But something is calling me, and I'm sorry. A castle in quicksand. I'm a fool with a bird in hand, straight in his dreamland. Even I don't understand, and I will glitter with envy of your new version without me. I'm only stupid, don't hit me. I'm trying to do the best I can. You're guarded, I'm leaving, so sad to Was it something she said? Maybe something he did. I could play along, look the other way. If there's something, baby, you should let them all know. It's not giving in when you're letting go. If you're thinking about taking the high road, I can tell you that you're doing it wrong. There's a better way of letting it all go. 'Cause you've been running from the feeling too long. Way too long. Did you let it all go? Should you let the girl know? Did she do you wrong? Need a lighter load? Were they holding you down? Was he yelling too loud? Did they break your heart back? Are you acting proud? If it's something, baby, you should let them all know. It's not giving in when you're letting go. If you're thinking about taking the high road, I can tell you that you're doing it wrong. There's a better way of letting it all go. 'Cause you've been running from this feeling too long. Oh, so you wanna paint a picture of every little thing that I did wrong. Now you're thinking about taking the high road. 'Cause the road you're walking's taking too long. It's that simple, baby. You should let them all know. It's not giving in when you're letting go. If you're thinking about taking the high road, I can tell you that you're doing it wrong. There's a better way of letting it all go. 'Cause you've been running from this feeling too long. Oh, so you wanna paint a picture of every little thing that I did wrong. Now you're thinking that you might take the high road. 'Cause the road you're walking taking too long.
for our virtual attendees will be starting in a few moments. We're just waiting for a few more people to make it into the room. Thank you very much.
Welcome back, everyone. Welcome back. Hope you can all hear me. We have a very exciting and robust schedule for this evening, leading up to our keynote fireside chat with Dr. Peter Marks from FDA. But before we get started, we would love for you to bring your attention to the screens again. We're going to hear an important message from Catherine Bryant-Knudsen. She's going to share with us a special message, and she is the founding president of the Speak Foundation.
We are on the brink of making the dreams and the prayers of so many come true. We are seeing hope for the first time, but it is just out of our reach. There are still no FDA-approved treatments for LGMD. Parents are devastated at the slow progress in drug development in a time where we are seeing amazing advances that are unprecedented in science. This community has come together over the past few years to raise its voice and inform drug developers and regulators of the challenges of living with LGMD, with the hope of making treatment options a reality. So many patients participated in the ELPFDD for LGMDs, the recent participation in the CBER PFDD for gene therapies, and the most recent publication of the Voice of the Patient report for the LGMDs. LGMD is a diverse group of muscle wasting disorders affecting both children and adults with a genetic root cause. Regardless of subtype, the LGMDs share many similarities. They are serious, ultra-rare diseases. They are progressive and debilitating, affecting muscles in the arms, hips, and legs. The LGMDs can cause cardiac and respiratory issues, and in some cases, even death. Today, I want to share important information and knowledge with the LGMD community, and this is the basis for our advocacy moving forward. We need to work together as a collective voice. First, we have a huge unmet need in LGMD for trials and treatments that slow progression. LGMD is very serious and it can be deadly. I have numerous friends who've lost their lives. Over the course of 15 years in this field, I have personally undergone the loss of ambulation. I've seen friends go from walking to a wheelchair and from independent breathing to a ventilator. Second, LGMD is somewhat unpredictable, with patient experiences being variable within the same subtype. In fact, we see even siblings with the exact same mutation progress at different speeds all the time. One person can lose ambulation at one age, while another with the exact same subtype can lose ambulation much later. Third, due to the small number of patients we have, we need creative approaches to drug development. Traditional approaches take way too long in a slowly progressing disease. Historically considered the gold standard, the randomized placebo-controlled trial model is a poor fit for the LGMDs. With such a very small patient population, there just isn't enough existing patients to power a randomized controlled trial with the rigor you see in traditional trials. Fourth, the very slow and irreversible progression of LGMD makes it very difficult to measure efficacy based on clinical endpoints like physical outcome measures. It would take years and years of waiting for physical outcome measures to show efficacy. Using surrogate markers such as protein expression allows us a way forward. When we see the protein expressed, we basically are seeing the evidence in a brilliant modern method. We are living in a scientific revolution. We all tip our hats to marvelous scientists who have changed the trajectory for so many. We've seen gene therapy and SMA. Babies that receive gene therapy and SMA are still alive years later. It was observed to work because SMA progresses so fast. With a slowly progressing disease like LGMD, we're going to be waiting years for physical outcome measures to show efficacy. At CBER's PFDD listening session for gene therapies, LGMD patients spoke about the value in slowing or stopping disease progression. They were all happy to see progression of the disease slowed, and they were happy to see treatments that would do this. Let's respond to this call to action. We have been given the flexibility already needed by Congress to give the FDA permission to ensure that the LGMD patient perspective and experience is accounted for when the FDA evaluates benefits and risks of any LGMD therapy. In the last decade, for example, Congress has continuously encouraged FDA to leverage accelerated approval to support the use of surrogate endpoints. As well, the FDA has the ability to allow trial designs that use natural history comparators. Finally, we need to ensure that drug development and review in LGMD is grounded in the latest science and understanding of this disease. To this end, the Speak Foundation intends to convene a scientific workshop early next year on February 8th, 2024, to bring all stakeholders together, including patients, clinician, industry, and FDA, to discuss the current state of scientific and clinical knowledge in LGMD. We're happy to announce that Dr. Peter Marks, who is our guest speaker tonight, has agreed to present at the scientific workshop, as well as Dr. Peter Stein from the FDA. Please hear us. The opportunity is now. The science is here now. Patients are in need now for all stakeholders to work together towards the goal of providing LGMD patients with a chance for treatment. We cannot wait.
We continue to thank Catherine for her fearless leadership of the LGMD community. We would also like to take some time tonight to recognize a world-renowned clinician and scientist who has decades of experience. This doctor has left a profound and undeniable impact on the LGMD community and broader muscular dystrophy community for his relentless dedication to research. He's published more than 370 articles in his career and authored several books with a focus on neuromuscular diseases and gene therapy. He has also been the principal investigator on numerous clinical trials, continuing to pave the way for innovative and much-needed treatments. This year, he was an instrumental part of the team that applied for, advocated, and received approval for the first ever gene therapy for Duchenne muscular dystrophy, Sarepta's Elevidys. This was a pivotal moment for gene therapy and a huge win for our friends in the Duchenne community and for those of us living with LGMD. Dr. Jerry R. Mendel has helped give us the one thing we will never lose, hope for a cure. We're honored to have this pioneer as our guest of honor this weekend. Living with LGMD 2A R1, I can personally attest to the impact that Dr. Mendel has had in my life as one of his patients. I felt a deep assurance, comfort, and peace knowing that I was being followed by one of the very best LGMD doctors. I trusted that I was always in the best hands and was being informed about the latest progress being made. Dr. Mendel brought expert care, compassion, and again, hope to his patients. Thank you, Dr. Mendel.
I have one more.
Oh, you want me to sit?
No, would you want to sit? Look at your chair.
It's called.
Learning of my son Jacob's diagnosis of LGMD 2D R3, where is he? There he is. In 2021, left my husband Josh and I scared, confused, and heartbroken. What did this mean for our sweet 5-year-old little boy who is seemingly so strong? What do you mean there isn't a treatment available for him? And what should we do next? Days of constant research continue to point us to one specific neuromuscular doctor in one location, Dr. Jerry Mendel at Nationwide Children's Hospital. From the moment we learned that Dr. Mendel would take Jacob under his care, our fear changed to hope. We felt assured knowing we were in the best hands possible for our son. With every visit, Dr. Mendel made us feel like more than just a patient. He took his time talking to us, was caring, and Jacob loved him. As a mother, the feeling that everything is going to be okay, if only for a moment, is priceless. And I know it's not just our family that Dr. Mendel has done this for. It's countless others for years. So tonight, we honor this dedicated man with another award here on the International LGMD stage. The very first LGMD Clinical Excellence Award. Thank you, Dr. Mendel, from the bottom of all of our hearts, for always pushing science forward, for the continual pioneering and research you provided the muscular dystrophy and especially the LGMD community. You are a true scientific rock star in our eyes. Because of your years of devotion, so many patients in the years to come will be one step closer to getting the critical treatment that they need. And thank you, Mrs. Mendel, for sharing your wonderful husband with our patient community for all of these years. As we all know, behind every successful man is a woman who has blessed him with love, support, and encouragement. Ladies and gentlemen, Dr. Mendel.
Thank you. I just have a few words, and one, I want to thank Rachel and Jessica, who I have dealt with personally for what obviously is a very successful conference, and the honor, it's virtually overwhelming to be in this position. I actually had to sit down in front of the photographer as I was marched up on the stage a little bit ago, and that was almost overwhelming. The courtesy, everything that's happened that has made this possible. One of the things I'd like to point out actually is that, well, one, I've been in neuromuscular research for 51 years. Started at NIH in 1969 when I saw my first muscular dystrophy patient. It's also important in this conference to realize that the very first patient who had gene therapy for muscle disease was a limb-girdle muscular dystrophy patient, and that was in 1999. So I personally have been at this for more than 24 years and devoted my career to neuromuscular disease gene therapy, and it's been very gratifying, and I appreciate the honor that is being bestowed on me tonight. So thank you very much, and I'll treasure this very much. Thank you.
Not so fast. In our next session, we will hear directly from people living with LGMD across various subtypes. This panel will be led by Kelly Brazo, founder of the Cure LGMD 2I Foundation. But first, we would like to play a few short videos from families of younger children living with LGMD to share their intimate stories living with this rare disease.
Let me tell you about my incredible 10-year-old son, Hudson. He has the kindest heart and the most infectious spirit. He loves playing video games and hanging with his best buddies. Hudson also happens to have limb-girdle muscular dystrophy type 2D R3. In hindsight, we began seeing symptoms of LGMD when he was a toddler, when he'd often complain about leg cramps. His doctors didn't have any major concerns back then, but at 8, after a growth spurt, we realized he was really struggling. Running, family walks, getting up off the floor and climbing stairs were, and continue to be, a big challenge for him. When we mentioned these issues at his well visit, his doctor suggested physical therapy. That very first assessment from the physical therapist set in motion a series of appointments and tests, ultimately confirming via genetic testing that he has LGMD2D. Since his diagnosis, we've learned a lot about this condition. Unfortunately, there's no magic fix for Hudson, no way to put the brakes on or slow things down. So our fight is all about physical therapy, aquatherapy, nightly stretches, and a weekly steroid treatment. These routines help Hudson keep his range of motion and hang on to as much strength as possible. And it's not just a physical boost, it's a mental one too, as it gives him a sense of control in this battle. While those tools are an imperative part of managing his disease, they are no match for the unforgiving progression of his LGMD. More than anything, we dream of a cure, or at least something to slow the progression. If there's a chance for Hudson to be part of a clinical trial for LGMD2D, we're all in. We would move mountains for a cure, not just for Hudson, but for all the people living with LGMD. It might be rare, but it's a part of the lives of some truly special individuals, my sweet boy included. Early intervention is key for kids like Hudson, who are just starting to explore the world. A cure would rewrite their stories, taking away the fear and uncertainty. It'd mean Hudson could live the life I envisioned for him when he was just a hopeful little bump in my belly, long before I ever knew what muscular dystrophy was. Please let us all continue fighting for a cure. Keep innovating, keep trying new things. We cannot give up until we see a cure for the nearly 500,000 people who are living with LGMD worldwide.
My name's Joe Dion. I'm accompanied by my wife, Courtney. Unfortunately, we have 2 young children with limb-girdle muscular dystrophy type 2C.
Last August, we learned of our son's diagnosis. Peter was 9 at the time, and, looking back, his symptoms probably started around 6 to 7 years old. Trouble keeping up with peers and running and climbing the stairs. But last summer, we really noticed that he was having trouble doing things that he loved to do. He's loved to fish with dad. Last year he reeled in his own 40-inch bass, something really, took a lot of strength and power to do that. And then this year it's definitely a struggle. And we noticed a decline. So we took Peter in for labs, and then there it was, a sky-high CK indicating some form of muscle damage. Once we learned that it was limb-girdle and that it was recessive in nature, that it could be present in both male and female, we had the gut-wrenching feeling that there's a possibility that our daughter Maggie could have the same thing. She was 6 years old at the time and exhibiting early signs that Peter had exhibited around 6 years old. She was toe walking. We decided to have her tested as well. We ran the same genetic testing, and it came back with the same genetic mutations, positive for limb-girdle 2C. She's 7 now. Every single day we noticed the challenges that Peter faces, and now Maggie is facing as well. Our goals would be to, number 1, extend their chance of survival. Number 2, we would want to improve their quality of life. It is important that they have the same opportunities for a chance of a treatment that other rare diseases have as well. There needs to be a shift. There are some challenges for developing these drugs for these ultra rare diseases. It's heartbreaking to watch children sitting and waiting and suffering. We appreciate lots of the thought leaders in this area that are trying to think outside the box, trying to create a new approach for these ultra rare diseases.
And we don't have time. We need fast action because we literally have a chance to help our children if we can get the ball moving quicker.
And every day is muscle lost.
So
Thank you for having me. I wanted to say thank you to Farron and to the Dion family for sharing their intimate stories with us. They are, I know it takes courage to share your story, and we have another panel of experts up here who are living with LGMD, so I want to introduce all of you. If you just want to share your name and your subtype to the community, and then we'll get started.
Sure. My name is Patrick Motion. I come from New Hampshire. I am 2ER4. A lot of fun to be with, and a Scorpio.
My name is Andrea Lane. I am from North Carolina. I have LGMD2A/R1, and I'm a Gemini, baby.
I'm Carol Abraham from Twin Lakes, Wisconsin, and I have LGMD2A/R1, Calpain 3 related. And I guess I should add that I'm a Pisces.
So I'm Peter Furring. I'm from California, and I have LGMD2C, and I'm a Scorpio.
I'm Jane Lockwood. I'm from Charlotte, and I'm a Leo.
All right. Well, thank you for being here. And many of you participated in our ELPFDD not too long ago. And one of the things that, the main concerns that was expressed during that time by our LGMD community was the fear of becoming a burden to our loved ones. How has that impacted you and your family relationships? How does that make you feel? And please share how that has affected you emotionally. Whoever wants to start.
I can begin. As someone who is dependent on someone for basically nearly every activity in my life, all of my ADLs, everything that's very personal and intimate, it's very hard to be dependent on other people. And not a day goes by that I don't think of the burden that I am to my husband, although he assures me he loves me and it's not a burden. It's one of those things that you just cannot overlook. I go anywhere with my friends have to help me. And it's very hard. It's hard on self-esteem, and it's just one of those things that never disappears.
Can I? Yeah. I'll add to what Carol said is the other part of the burden is not knowing what the future holds. You know, once upon a time, all of us up here, we weren't a burden. And as time has gone by and muscle loss happens more frequently, things get more difficult. So now being a burden is a different definition than it was last year, 5 years ago, 10 years ago. And the real fear is what's the burden going to look like in the future? Because as things start to go, you just become feeling more of a burden. So the self-esteem, definitely I echo Carol. But it's also thinking about what's going to happen.
Yeah. Does anybody want to add?
I can. So I'm, of course, the youngest person on this panel, being only 16, and I'm hopeful that I will be able to self-sustain to an extent, but I know always that my family will be there for me if I need them at any point in time. They'll help me. But I also want to, I'm hopeful for a cure for muscular dystrophy or even something to just stop progression to allow me to keep some of my independence throughout the rest of my life.
I think a lot of people have spoken about family, and we're fortunate. I'm in the same position that my family is very caring and loving and supports me always. I joke that I live with my 2 roommates, mom and dad. Met them 27 years ago in the hospital. Real love story. But they take care of me, and then I also think that it's really important to have open communication with your friends who, may not necessarily understand the progression of this or the ins and outs of the disease. And so I always try and be open with my friends, and they're open with me, and they're very honest about, if they're going to go on a beach trip or to the mountains or something, and it's something where the stairs doesn't have an elevator, or the house doesn't have an elevator and there's too many stairs, then I've had discussions with them about, hey, I know that I can't go on this because it'll be too much, but I appreciate it if you let me know that you're going before. And I think that it's just important to have open communication so that you don't feel like you're always a burden on people and hurting people.
Yeah, I think I'll just add, I think it's hard because our culture, particularly in America, I think is very individualistic. So we really value independence, and that's what leads to these feelings of feeling like a burden for your loved ones. And so I think in living with this disease, I've tried to kind of deconstruct some of those thoughts and understand that my relationships with my friends or my family, like it's gonna feel different. And how can I try to have some reciprocity in those relationships, but know that it's gonna be reciprocity in a different way. And I think in that way, maybe you start to feel less like a burden.
Thank you. That was excellent. So if a treatment was to become available that was to stop the progression of your disease, but did not necessarily improve your condition, would that be considered a success to you? And how would you feel about that? Some of you mentioned a little bit about that, but do you want to elaborate?
I can start. I think that would be a huge success because I currently can still use my arms and I can walk short distances. So I would appreciate being able to continue doing those things. And that would, in my opinion, be a ginormous success because I can be more individual and feel almost less like a burden, going back to the last question.
I am currently in the ML Bio trial, clinical trial for 2I. And if you were at the Lunch and Learn today, they talked about how that treatment has, it's slowing the progression, which is wonderful. And I feel like I've seen that. So I don't necessarily know if it's scientifically available, that data yet, but I feel like I can see a difference. And I think that that's something that has made a huge difference in my life.
It would be a dream come true if I could just stop this progression. I've had weakness progressing for over 56 years, and I'm running out of ways to adapt. It would be beyond belief if I knew that the progression would just end and I would have some stability.
Yeah, I definitely agree. I think the progressive nature is just what makes it so challenging because you're constantly having to adapt and seek out new resources, new tools, new equipment, everything that goes along with that. And so if you can just stop the progression and just be able to have an environment that works for you, obviously it's still challenges, but I definitely think that would be a success.
After 56 years, I can say I'm running out of ways to adapt. I'm running out of equipment. I need something to stop the progression.
I'll push the envelope further and say, too, that the longer you live with this, because I'm going on 40, I don't even remember, 40 years at least, I'm running out of patience, and I'm running out of nerve. Again, it would be a game changer because we could also then plan for the future. Not knowing what that looks like and what other services we're going to need, treatments that we're going to have to make available if it's more power equipment, that's the most terrifying part is the unknown. So if somebody could look me in the eye and say, this drug is going to keep you level for a while, that would be a game changer. Yeah, I don't think any of us are expecting that we're going to get up and walk. I don't think any of us really value that. I don't want to speak for everybody, but just stop it in its tracks, that would be wonderful.
That makes sense. So how hopeful are you? Tonight we've heard a lot of the word hope, right? So there was a big theme with our conference, Be the Hope for the Cure, right? And how hopeful are you that you'll see a successful treatment in the coming years? And how are you coping in the meantime while you're waiting for a successful treatment to be approved for LGMD, for your subtype?
I think that conferences like this are extremely important to get to see the science behind what's going on. My parents started the McCall-Lockwood Laboratory about 20 years ago, and we have had 2 therapies come out, a gene therapy and then the ML Bio trial, and seeing those progress over the years and to the point where it's finally at a clinical trial is really hopeful. And I think that that's something that I believe will definitely be maybe not available on the market in 10 years, but something that will be, the clinical trial will be progressing and we'll have a better further step towards a cure.
I would say I'm hopeful, but I'm a little frustrated that there's been so many delays in treatment. But I am hopeful that there will be a treatment that will come sooner rather than later. And I think the way I cope with it is like I have to do things, I have to deal with it. It's a part of life, but it's something you just have to overcome.
For me, it's very exciting, the first time in my life to see so many treatments entering into clinical trials, and that there's hope like there's never been before in the LGMD community. But at the same time, it's so frustrating that, as an example, for my form of limb-girdle muscular dystrophy 2A, just sits in preclinical phase of a pipeline. I wish that there was a way that we could accelerate this process in some ways. I'm afraid that by the time something comes available, that I won't have enough healthy cell muscles to be able to treat. And that's the worst feeling possible, to think that maybe in my lifetime there could be a treatment and a cure, but then to be so far progressed that it wouldn't be available.
That's a tough one to follow.
Yeah, I'm sorry, I can't.
It's the truth.
Yeah, I know.
Do you want me to?
Go ahead, Pat.
No, listen, that's what we're up here for, right? Truth. And you want to hear some harsh words. I'll flip it the other way, though, and hope is this room. Hope is all the people that made this conference possible. Hope are the people that are working on this and have been working on it and dedicated their lives. Hope are the people that deal with the government. We'll just leave it at that. But I've been doing these kind of conferences for 20 years, and I've made friends that I have for life. And I tell everybody that doesn't understand, the silver lining of this is it's the best family that you never want to belong to. You know, the people I'm having dinner with, I just met recently, and I said, you know, it's very nice to be here and not have to talk about muscular dystrophy. I know that's the main concept, but we don't have to explain it. You don't have to say to somebody, oh, I had this, and then they say, oh, my aunt had MS, and you say, oh my God.
The worst.
But, right, yeah, we all know that without, no. But, so, I mean, the hope comes from everybody in this room, and I share with Carol, you know, I just turned 50, and yeah, it's getting more frightening, but the other part of me thinks, all right, then maybe, you know, I help the people that I just saw in the video. I help the younger generation that has the coolest hair on the stage. We're gonna do that too. So it's a struggle every day between being very selfish, but also trying to be selfless and saying everything that I do, every natural history study, every time I'm poked, I'm prodded, blood work, I'm adding to the data and the pool of data that's needed because we're so rare. Yeah. All right.
You want to say something? No. You're not ready yet. Okay. Is there anything else anyone would like to add? Any other things? I'm out of questions, but you are welcome to add more, or if you want to just go eat your dinner, we can do that too.
I think going off of the coping skills and trying to look at things positively, I know I went and saw Sammy in the Teen Talk earlier, Sammy in Brooklyn, and it made me think about how when I applied to colleges, everybody's always like, oh, what is my college essay going to be? Well, this is your college essay. It's really easy. So just flip it. You have a college essay. So, and siblings, that goes for you too. So
yeah, that's so true. That's so true. Well, thank you so much for sharing your stories. And it is not easy to get up here and share some of your most intimate fears and challenges, but also your hopes. And we all do have a ton of hope, but we also all share the same resounding story that, you know, things can't happen fast enough for our community. And we are grateful that you're all here to help us, you know, have hope. And we'll take some of this with us for sure.
We're also extremely grateful for the clinicians and the researchers who know the urgency of our community. And we know that you're working hard. And we are extremely grateful for all the work that you are putting into our community.
I'll just end with stay loud and stay active and keep the network going, keep everything going on social media. We all have to crowdsource problems. We all have to speak together with each other more. Speak Foundation, see what I did there? But everybody, we have to continue to work together. So we'll be here all weekend. Please tip the wait staff. Come see us whenever you want. Thank you for having us up here.
All right.
Thank you.
I'll lead the way down.
Thank you.
I'll let that come up. Next. So, you know what, I don't think so. We'll have. I have a table to hide behind. I like that. I'm more comfortable behind a table. We're ready? Good? All right. All right, can we have your attention, please? Okay, Brad. I don't think his mic's on.
It's part of our.
Hold on, I don't think your mic's on.
Am I? Yeah, okay.
We're good? All right, go ahead.
Okay. Okay. For the next part of our evening, we're honored to have Dr. Peter Marks as our guest. Dr. Marks is director of the FDA Center for Biologics Evaluation and Research, commonly known as CBER. This is the area of the FDA which regulates such treatments as gene and cell therapies, protein therapies, and also vaccines. Dr. Marks received MD and PhD degrees from New York University and did his medical residency training at Brigham and Women's Hospital in Boston. Since then, he has held positions in both academia and pharma industry before joining the FDA in 2012. And he's been the director of CBER since 2016. So please welcome Dr. Marks. So to start off, a lot of the treatments under development for limb-girdle muscular dystrophies are gene therapies, particularly gene transfer therapies. And I wonder if you could sort of go into some of the comparison between the drugs like these versus more traditional drugs from both a development and a regulatory perspective.
Yeah, so thanks. First of all, thank you very much for having me here tonight. And I have to say, you're truly an amazing group of people, so thank you. I think some of what we've come to learn over the past couple of years is we've gone from having no gene therapies approved prior to 2017 to having now a total of 15 products on the market. and a bunch that are almost there as well. Small molecules are, they generally have been developed over time. They cost a fair amount of money to develop. They have toxicities that are sometimes hard to know up front. And so it can be very challenging to develop small molecules for diseases. The good news when you get them developed is they're usually not incredibly expensive, but that's not always the case. Gene therapies have going for them the fact that if you know what gene's missing and you know what needs to be put back, there should be a rational way to make them. So that's the key thing here is that right now for many of the gene therapies that we see, they are essentially a backbone. The best thing is the analogy is like a razor handle to which the gene of interest is like a razor blade that's put into it. And we're increasingly realizing that, you know, there are some fundamental differences here that make these gene therapies really a little different than other products that we deal with, like the small molecules, because the toxicities are shared essentially usually with the razor handle between the different products. Not invariably, but it's a pretty good bet. And that may mean that we can use some tricks to hopefully move forward more rapidly and have more gene therapies approved in a very rapid manner. And probably we can discuss as we go along some of the ways that we think that we'll be able to make more haste through getting gene therapies across the finish line.
Okay. Now, as a follow-up, I have what little I know about gene therapy manufacturing is that that's also a big challenge both to developers and FDA.
Right. So gene therapies, the gene therapy vectors that are made today, they're either viruses like adeno-associated viruses. For cell-based gene therapies, they're lentiviral vectors. And now for the genome editing products, they're going to be very interesting mixtures probably of potentially of DNA and DNA RNA mixtures or RNA and proteins packaged in liposomes, essentially a delivery vehicle. This is going to require a lot of work to be able to make these in a facile way, because right now they're made very much in a boutique manner. It's not like you can go to a machine and crank these out. They're often made essentially in academic laboratories or in manufacturing facilities where there's a lot of hands-on work. And that's led to very high costs. In many of the gene therapy trials, it costs, just the cost of the product itself is $100,000 to $250,000 per dose to make the product. And so one of the things that we're very interested in doing at FDA is working with the community of manufacturers, nonprofit organizations, and we're working in a partnership with the Foundation for NIH called the Bespoke Gene Therapy Consortium, trying to figure out ways to semi-automate the production of gene therapy to try to bring down the cost and speed up the production. Because right now, some of the ability to get gene therapies into trials is not just a matter of regulators being slow. It's the fact that people can't get these made at contract manufacturers fast enough to get into people. So hopefully we'll overcome this in the coming years. I think people are finally realizing that manufacturing is the challenge that it is. This is an interesting thing. It shows you how mistakes can be made. About 5 or 10 years ago, when we were first starting to see gene therapies increase in number, a lot of people said, oh, don't worry too much about manufacturing. It'll all take care of itself. It'll be just like monoclonal antibodies. that was a real mistake. And when you think back to it, the people who were thinking about the business models should have realized, and we should have realized, that they're very different. Because gene therapies actually, if they do their job correctly, they're one and done. Whereas these monoclonal antibodies are continue to be produced. So in some ways, the challenge of one and done creates an issue here because it's almost so good.
So LGMDs are considered rare or even ultra-rare diseases. And what challenges would you say that this creates for drug developers and for regulators?
Right. So I think there are a couple different things, and at the risk of kind of going through more than you'd like to hear about, but we have dinner, so we can go on. And like sometimes I say, oh, we'll order a pizza, and I'll tell you about it. So there are a couple different problems. One is when you have rare diseases, it's hard to, especially ultra-rare diseases, it's hard to figure out what endpoints we're going to use to get things over the finish line, because ultimately we have to show that something is showing some type of benefit in order to say something that's going to get an approval. There actually is an approval standard that basically says we have to be convinced that the products got efficacy of some type and that it's safe. And the good news is that in that regard, to say that something has efficacy, and this was mentioned already before, Congress has given us some leeway. We have the leeway of being able to use something called accelerated approval, where rather than using a clinical endpoint of how someone feels, function, or survive, we can actually use either a biomarker, which is a laboratory test, or an intermediate endpoint, which is perhaps not a final clinical endpoint. And for gene therapies, they're very well suited to using accelerated approval. And I think especially when you think about LGMD, you're talking about something that may be very well suited, because you can't wait 5, 10, 15 years for a typical clinical endpoint. What you'd like to do is look at something that you can measure in a year's time to say that something right happened when you gave your gene therapy. And that means that it may just simply be looking at whether a year out you have good protein expression, that we know then that protein expression in some model system or in individuals who have different levels of expression, which we know happens in the community, that level is either associated with either normal or only mild problems. And so if you can show that you can match up something like that, we can use our accelerated approval pathway, and that will hopefully get us over the finish line. And then the final thing that I'll say about rare, ultra-rare diseases is something that we have as regulators as a problem globally, which is that our goal, although we are the United States Food and Drug Administration, it's not lost on us that gene therapies are probably best, they're the best hope for patients with rare diseases, not just in our country, but globally. And so the hope would be that we can help get to better regulatory harmonization around the globe, so that when we have something that we feel comfortable has come across the finish line in the United States, it's easier for it to get across the finish line in other regulatory environments in Europe or in Asia, and vice versa, because that will actually, it may sound trivial, right? But expanding markets like that help actually get developers to be more excited about going into rare diseases. And though I tend to be an idealist, so I would say, look, the developers should be doing this because it's the right thing for public health. But at the end of the day, we have to be realistic. The way we will help make sure as many people in this room get cures potentially is by having a robust commercial enterprise that makes people want to go into this area. And if we can help open up markets around the globe. It's an incredible win-win. Patients get access. That's what I care about, and the companies have a larger market for their products.
I was going to.
Thank you. You largely have answered like the next two questions that Kelly and I have to ask you. But so I wanted to go back to, you gave a keynote presentation at the Muscular Dystrophy Association conference earlier this year, and you actually spoke about some of these issues, having to do with making, using accelerated approval for gene therapy. I guess one question I have is, because LGMDs are ultra-rare diseases, and unless you just say, well, they're missing this protein, if we put the protein back, that's the surrogate measure. A lot of times there aren't well-established, biomarkers or surrogates. The thing is, we've never been able to treat these diseases before, so, how do we know what actually correlates with improvement?
Yeah, so I think this is where we feel like I think you use whatever tools you can have at your disposal, and I think it's a combination. In some cases, it may be a combination of what we can get out of animal models and what we can get out of natural human experiments. So I'm a hematologist oncologist by training, and in hematology, there's a very analogous situation where we can measure certain genetic diseases have varying levels in different patients with the disease. And some of them, the varying level is just so modestly off that they're basically okay. In an occasional, they may have an inconvenience occasionally, but if we could get to those levels for everyone, we would know we would be okay. They don't, for instance, in a bleeding disorder, they will only bleed in a very certain circumstance, and we can deal with that. And so by getting to that, if we can take people who are at levels that bleed spontaneously and get them to that level, we can know that just by changing the level, measuring the level of the protein expression of the gene therapy, that's good enough. And I think the same thing, we try to use all the levers we can here. Some there'll be animal models, some will be the human, and some will be complementary data, and we try to find a way to put together what can convince us that there's benefit. It's really important that there be benefit, because I think we don't want people to have, the most terrible thing that we can do as doctors is give people a medicine that doesn't actually work, right? It's false hope. And it takes away opportunity for products that might actually work. So I think as long as we can convince ourselves, and I think that's the exciting thing about working in this area, is to actually leverage all the science that we can. I think we'll hopefully be able to get there somewhat faster. It's going to mean taking some risk that we might be wrong. But I think what we heard from this panel here is that if there's a certain risk of that, as long as it's not a wildly crazy risk, as long as it's a very calculated small risk that we're not right, people probably will understand that we need to try what we can try to move things forward. And it's going to be uncertain, because for some of these things, I think it was beautifully said, right? If you give a gene therapy to somebody who's had a disease for 40 years, it may not, it's not likely. I mean, we'd love it if it could happen. Believe me, I'd love it if it could, but it's not likely to make them be able to get up and walk. But this idea of being able to stabilize things is a very valid endpoint. The problem is those are things that can take a while to show, and it's okay, as long as we can convince ourselves with a marker that it's likely that when we get the data in 5 or 10 years, that we're going to get there. I don't have any problem with having things sit out there with accelerated approvals for 5 or 10 years. We could have a whole, I go to, in other more common diseases, we go to debates about whether accelerated approval is a good or bad thing. And I basically say, just stop it, because in certain diseases, accelerated approval is going to be the only way we get there.
And you did mention a bit about the safety. And what is the FDA looking for in terms of safety and efficacy of a treatment in order to approve it? For example, how does the needed level of confidence depend on the disease, the treatment modality, and the patient preferences?
Right. So, I mean, this is really an important thing that has, I'll just back up and say something. We were talking before this about, earlier on today, a little bit about what people who work at FDA are like and how things have changed over the years. 2 decades ago, a lot of what we did at FDA, or 3 decades ago, were things for large populations where there was a lot of risk aversity, and probably justifiably so, because for people who are going to take a medicine for high cholesterol or high blood pressure or who need a blood thinner, we give them to so many people that if they had a terrible adverse effect, that's not a good thing, right? On the other hand, for a rare disease community, the risk-benefit calculation is very different, because when the outcome is virtually certain that it's going to be bad of the disease itself, people are willing to take a different risk of a therapy. Now, we can't be wildly crazy and give unsafe therapies. On the other hand, if we can define the risk so that people can make an informed decision, I think we know that there are probably, for instance, with some of the upcoming gene therapies, there's probably going to be very small risks, calculated risks, that they could cause at some point a cancer in the future for some of them that are in development. But the chance that they're going to bring benefit without that is much, much, much higher. So as long as people can understand what's the potential benefit and understand the risks, I think that risk-benefit calculus can be understood. And I have a lot of experience with that, because as a doctor who used to treat leukemia, it was a constant conversation that we would have. There was if we don't do anything, we know what's going to happen, right? Very bad outcome. If we do something, there's a small risk of a bad outcome, but a reasonably good chance that we could have a very good outcome of cure. So I think patients are in the best, and their families are in their best position to help make that decision. And our job is just to make sure that when we put therapies forth, they have at least, they have to have a certain bar. They have to meet a certain bar so that when that discussion comes up, it's the risk benefit is at least in a proper place. And what does that mean? It can't be 50-50. You know what I mean? That's not good enough. Oh, 50% chance it'll help you, 50% chance it'll hurt you. On the other hand, if it's 99-1 or 98-1 or something like this, we can live with those. And I think it's because we're just transparent about what the risks and benefit are. And I think people might, they might decide at different stages of their disease, this is worth it or this is not worth it, knowing what the alternatives are.
So how does the fact that, as it stands now, gene therapy is a one-time treatment, how does that affect these decisions of risk and benefit?
Yeah, so, I mean, one of the things about our current gene therapies is that we'd like to, this is a challenge because with AAV, the adeno-associated virus, one of the common vectors being used, once you give it once, people currently make antibodies, and it's very challenging to give it a second time. People are working on dealing with this. The next generation of gene therapies coming out that may be based on these lipid particles, they may be a little easier to give over again. But this, the hope here with these is that we get it right, because we like one and done to be the case. But I think whether they cure or whether they provide long-term benefit, it's still, I think at this point in time, they're both acceptable endpoints, because I think, again, whether it's cured or whether it's 5 or 10 years of benefit, for somebody who's losing function, if we could stabilize function for 5 or 10 years, that's probably an okay endpoint.
Don't let the perfect be the enemy of the good.
That is exactly, I think that's exactly right. We can't let perfect, and we tell this to our reviewers, don't let perfect be the enemy of good, because we tend, again, regulators are risk averse. It's just the nature of things. We are risk averse people. You should see my financial portfolio. It's completely risk averse. And Uncle Sam benefits from it because it's all in Uncle Sam. But we are a risk-averse bunch, but nonetheless, if we're not willing to take some risk here, we're not going to help people. And so that's, I think, the impetus to bring together a group of people where we are changing the culture at FDA from people who are highly risk-averse to people who are willing to take some risks, knowing that they could occasionally be wrong, because that's the only way they will help benefit public health. And so I think one of the great things about, just to come back full circle, patient-focused drug development meetings, the patient voice, I think probably the biggest help it gives us, rather than changing the bar of where we're going to actually approve things, is actually in helping our reviewers to actually understand what it's like to live with these diseases, what it means, what it would mean to have a therapy, so that they can feel the urgency that really needs to be there, and that makes a huge difference. So for those patients who have met with our reviewers and who have the opportunity to do so in the future, thank you for doing so, because it really helps them.
So one thing that's of great interest to the patient population is, when a drug is approved, will I fall under the approval criteria, and will before it's approved, will I meet the criteria for the clinical trials? So I wondered if you could comment a little on sort of, how. What determines the broadness of the approval or of the clinical trial enrollment?
Right. So this issue of how drug development is and gene therapy development is a little challenging in that regard, because at FDA we can give advice to companies, but it's in general they decide who they're going to put in clinical trials. That said, I think we are taking an approach in gene therapy now that's very similar to one that we have in cancer medicine, which is to say, look, try to make the entry criteria for clinical trials as broad as it can be. And even if you have to enroll some people who might not be able to be part of the primary analysis population, at least find a way to be able to treat more individuals to enlarge the safety data set, particularly in ultra-rare diseases. And ultimately, I think the reason why we like accelerated approval endpoints is that hopefully we can then, when we give an approval under accelerated approval, give it for the widest population that we can, knowing that the hope would be that if we see activity in one part of the disease, that's going to translate into some type of activity in another part. In other words, if we see babies that don't come down with loss of function, it's probably then, and we see others who don't have the decline in function, it would be hopeful then that we can at least stabilize older people. So that's I think, where you'll see us go. Again, will probably mean that people will be in long-term follow-up studies. It probably means that we'll be using real-world evidence to follow people over time. But that's just the nature of, I think, what we're going to have to accept for gene therapies for diseases that have a very slow natural progression, because otherwise, if we try to do, we know that the ecosystem, venture capitalists have an even shorter attention span than I do, and that's bad. And so they want the answer very quickly. And so we have to help them get products across the finish line, and then that's okay. If we can get them over the finish line in a year or two in a clinical trial, then we can follow people for years and document clinical benefit. That's okay. And I think some of these things will become obvious with time anyway.
That's great. That's so good to hear. And we heard from so many people that are excited to, we have a group of people that are willing to participate in natural history studies and obviously clinical trials too. And to say that we can try to encourage industry to broaden the patients that can get enrolled. One of the biggest things we hear is, yes, we all have hope, but there are also people who are losing hope. And I've heard our patient community members say, well, it's probably too late for me, but hopefully, we might see a cure happen in the next generation. And that's so heartbreaking to hear. So I'm so glad to hear you say that you're encouraged.
And that just takes us back full circle to this issue of manufacturing, because if some of the manufacturers, they'll say, well, we don't want to enroll, we can't afford to enroll more people. And if we could get the manufacturing costs down, hopefully they'd be able to enroll more people up front. It would be good for us because we'd have larger safety data sets, and really good for patients because I think, again, there are some people who, again, if you're 50 years old with a disease, you might want to take your chance now rather than waiting 5 years for a better vector or a better gene therapy.
I think for LGMDs in particular, part of what will help will be having better outcome measures for non-ambulatory patients.
That's right. And I think that's something that the medical community will work towards. And I think we're very open and willing to work towards this. That's one of the things that I think Congress has essentially instructed us to do, to try to work with patient groups, advocacy groups, et cetera, to try to come up with better outcome measures in a variety of rare diseases.
Yeah, that's good. Thank you. I think
okay.
that's
unless you have any final comments, I think we're out of questions.
So that's very sweet. I just want to say thank you all. I think we don't have all the answers. I think we're still early on in the days of gene therapy, but I think we are starting to see us come around a corner where I think we'll see the pace of progress really accelerate as we realize these therapies aren't small molecule or even protein therapies. These are kind of a new platform for hopefully correcting things that we can move more quickly through than we have in the past. So hopefully in the not too distant future, we'll see more progress in this area. But thank you so much, and thank you for all you do.
And thank you.